So question 1 was on polar bears. It says polar bears are highly adapted to cold climates around the North Pole. Brown bears, black bears, and pandas are found in warmer environments. Researchers collected mitochondrial DNA sequences from several populations and constructed a phylogenetic tree to represent their evolutionary relatedness. Okay. So for A, it says, use the phylogenetic tree in figure 1 to estimate the age in hundreds of thousands of years. So if we look here, hundreds of thousands of years for the most recent common ancestor between all brown bears. So this is a brown bear here, this is a brown bear here, and this is a brown bear here. So we trace them back, and that gives us this point right about there, which we're going to call 325,000 years. I think that's fine. There's probably going to be quite a big range for this. So we'll call it 325,000 years. And that was a estimate. Right, so that's 1 point here. So there's a 1 check there. And then the second part said to identify the population of bears to which polar bears are most closely related. So Euro brown bears and polar bears are the most closely related because they have the branch that's the closest there. So we'd say European brown bears. That's another point there. And then the last part of this question was identify 2 populations whose positions could be switched. Okay, so anytime you have the end of a phylogenetic tree like this, so this branch here or this branch here, those can rotate on their branches. So we could ascend European brown bears and polar bears, or Western North American brown bears and Asian brown bears. So we'll go Western, NA, and Asian bears. That was our third check. So part A was worth 3 points then. Part B says, construct a cladogram on the template to represent the model of evolutionary relatedness among the bear species based on the differences in the list protein sequences. Circle the position in the cladogram that represents the outgroup. So you can see that in this diagram here, we've got panda bears being really different from everything. 33, 34, and 40 differences. So we're going to put pandas up here, and we're going to call the panda the outgroup because it's got the most differences. Okay. Then we go to the next most differences, which is the polar bear that's got 7 and 8. So that goes there. And then it doesn't matter if you put brown here and black at the other place. The brown and black ends can be rotated on their node. Right, so you could have had panda polar black brown or panda polar brown black. Either one, doesn't matter. Part C says, a student claims that the mitochondrial DNA sequence comparisons provide more accurate phylogeny than of bear species than list protein sequences. Provide one piece of reasoning to support the student's claim. Okay, this question is going to be a lot of different possibilities, right? So one piece of reasoning for this could have been so many different things. I think a really good piece of reasoning that you could have used is that since this is using differences in amino acids, since the this chart up here uses differences in amino acids, and the other chart, the phylogenetic tree, wherever that is, here it is. Phylogenetic tree uses differences in DNA sequences. This sequence, or this tree shows all mutations, including silent mutations, whereas this one only shows missense and nonsense mutations, right? And so we get a better idea of the number of mutations. You could have also said that like mitochondrial DNA is more highly conserved than autosomal DNA. All kinds of different things you could have said here. I think there's a ton of different options. I'm just going to say that the silent mutations not shown on on list diagram, or something like that. Okay, so that's my point for that. 1 point there. This was 1 point for doing the cladogram, and then 1 point for circling the out group. And then D says, describe how the mutation of the list gene became more common in polar bear population. If the list gene were the only determinant factor of fur color, predict the percent of white offspring produced by the mating between a polar bear and a brown bear. Oh wait, no, sorry, we forgot D. D says, a researcher genetically engineers a mouse strain by deleting the mouse list gene and replacing it with the polar bear list gene. Predict the most likely difference in the phenotype. Well, what did it do to polar bears? Made their fur white. So we would guess, we'd predict white mouse fur. And then the second part is justify the prediction. Polar bears, mice, and humans are all mammals. And list has a similar effect on all mammals based on bear and human data. Something like that. Then E says, so that was a predict and justify, first of all, so those 2 points. Describe how the mutation in the list gene became more common in polar bear population. If the list gene were the only determinant of fur color, predict the percent of white offspring produced by mating between a polar bear and a brown bear. Okay, so why did it become more prevalent? That was because of selective pressures. So selective pressures gave white polar bears an advantage. So you could have said something like camo here in white environments. And then the second thing was the percent of offspring. Now, I think that they're probably going to accept another answer here, but I think the best answer here is to have the polar bears be homozygous recessive and the brown bears be homozygous dominant. And so it would result in 0% for that. 0%. And the reason why I think it's 0%, that you might be able to get away with 50%, but the reason why I don't think it's that is because if brown bears were heterozygous, if that was a really common genotype for brown bears, then you would expect 25% of all brown bear offspring to be white, and that is not the case, right? So I think this is the best answer for that, 0%. What's up?
For the list gene, could we say that the list gene coded for white fur polar bear, so therefore the mice would have white fur?
Sure. Yeah, I don't have a problem with that. All right, so here's my terrible drawing of the question 2 thing about the interleukins and stuff, right? And so anytime there's a parentheses here around something, that represents the inactive form, and then the non-parentheses represents the active form. Okay, so the question is talking about these pathogenic bacteria that go into cells and replicate, and then they spread to other cells, causing illness. And so, question 1, or question A of part A of question 2 rather, says describe the effect of inhibiting step 3 in the formation of the pores and on the release of interleukins. Okay. Well, this was step 1. This was step 1. This was step 2, and this was step 3. They were labeled on the actual exam. So, this is a pore. And step 3 is the part that makes the pores. So if you inhibit step 3, it inhibits the formation of pores. This is the part where the interleukins are made and released, and that's step 2. Step 2 becomes before step 3, and therefore it should not have any effect on the interleukin release, but it prevents pore formation, right? So we would say prevents pore formation. No effect on interleukins. Okay? I guess it's probably effect, huh? No effect on interleukins. All right. So then that was 2 points there. Boom.
What if you said that it would like double the interleukin production because it said like that it could cleave 2 proteins before it was used up, and since they wouldn't be cleaving the stuff to make the pore, it would double the cleaving for the interleukins.
That is an interesting take on that. One that I had not considered. Let's see. Inhibiting step 3. Where do you see that?
It says like caspase 1 or caspase 1 can cleave 2 other proteins.
I think it says 2 other proteins, meaning like it can cleave the whatever the thing was called. It could cleave the gastromin and the inactive interleukins, but I don't think it's meaning that it can only do it twice. I think it's just, I don't think it's saying it's used up afterwards.
Since that doesn't contradict the other.
Yeah, I think you're probably still okay. Yeah, I think that you'll probably still be okay. All right. So that was 2 points there. So 1 point for pore formation and 1 point for no effect on interleukins. The next question says, make a claim about how cleaving inactive caspase 1 results in the activation of caspase 1. A student claims that the pre-infection production of inactive precursors shortens the response time. Provide one reason. Okay, so you can see that for whatever reason, there's like a part on inactive caspase here, and then you remove that when the infection happens, and that causes for caspase to be able to do something. So in this case, we could say something like the shape of caspase binding site. You could have said active site. Changes or is made more available, like it like opens it up or something like that. What's that?
Can you talk about like
Allosteric?
Yeah.
Yep, that seems fine too, I think, in this case. Yep.
So is it okay if we just said cleaving caspase 1 remove the inhibitor?
Could be, yep. Could be removing an inhibitor. Yep, that's fine too. So then the other thing was, it said if we pre-produced a bunch of inactive interleukins and inactive gastramines, would that increase the rate of response in the bacteria? And the answer to that is absolutely it would, because if you don't make those, the first step to the bacterial or to the immune response has to be producing them, and protein synthesis takes time, right? So we could say protein synthesis takes time. So point there, point there. So we're up to 4 points. C says a student claims that the NFKB protein is located in the cytoplasm until the protein is needed for transcription. Justify the student's claim with evidence. Identify the 2 fractions where N-terminal gastramine would have been found in the cells. Okay, so we had this chart here where you had the whole cell and then you had fractions of the cell, and then it tells you what else was there. So this said that it was used in the Krebs cycle and it was found in fraction 1. That means that that fraction 1 was showing us stuff that was from what part of the cell? Where does the Krebs cycle happen? Mitochondria. Okay. DNA polymerase, that's found in the nucleus. So fraction 2 was from the nucleus. Glycolytic, this is a glycolytic protein, so it's part of glycolysis, which happens in the cytoplasm. And then this was the Na plus K plus ATPase, or the sodium potassium pump, which is found in the plasma membrane. And so then it is asking about NF-kB protein, and it says it's located in the cytoplasm, right? So can we support that? Did we find NF-kB in the same place as the cytoplasm? We sure did. Right, so we can say that NF-kB was found in the same place, or was found in fraction 3, which also contained a cytoplasmic protein. What's that?
What if we said proteins are synthesized in the cytoplasm?
I don't think that's enough, because you needed to justify the student claim with evidence.
Okay.
So we say NF-kB found in the same area as the glycolytic protein. Glycolysis happens in cytoplasm. Okay? And then the other thing said identify the two fractions where N-terminal gastramine is found. So N-terminal gastramine would have been found in both the cytoplasm and in the plasma membrane. So that would have been number 3 and number 4. So that would have been, check for that. And then D says, explain how gastramine pore formation and interleukin release contribute to an organism's defense against a bacterial pathogen. Okay, so formation of the pores does what? Wait, did we skip a thing?
Yeah.
Did it again. Okay. Describe the most likely effect on gastramine pore formation on water balance in the cell in a hypotonic environment. Okay, so hypotonic means that there is less solute concentration outside the cell, and so the water is going to flow into the cell. Okay, so we'd say water flows into the cell at a greater rate. Okay, that's checked there. And then E, we forgot about, E says, explain how gastramine pore formation and interleukin release contributes to an organism's defense against a bacterial pathogen. Okay, well, water flowing into the cell does what to the cell? Blows it up, right? So the cell is lysed. So it breaks apart, okay? And if the cell gets broken apart, that means that that bacteria can no longer reproduce inside the cell, preventing bacterial reproduction. And then interleukins are released, increasing the immune response. I think you could have said increasing the immune response, or in this you could have said triggering here too. Triggering the immune response. So 1 here and 1 here. So that's a total of 1, 2, 3, 4, 5, 6, 7, 8, 9? Did I miss one? 1, 2, 3, 4, 5, 6, 7, 8, 9. Where did I miss one? So we should have had describe the effect. Oh, and the release of interleukins. So two points for question 1. We got that, or part A. Make a claim, provide one reason. So that should have been 2 points for B. 1, 2. C, identify 2. So actually this was 2 points. Gotcha. Okay. So that was question 2. Question 3. Fun question. We had our aquarium set up. Right? It says, use the symbols below in a template of aquariums to demonstrate the experimental design for testing. The researchers claim that mobile aquatic invertebrates can pollinate seagrass in the absence of circulating water. Draw the appropriate symbols. Okay, so we had, in the control, we had the male and the female plants. And then the experimental, we had the male and the female. And then we also had the mobile invertebrates. That was hard. So that was check 1, check 2. Hopefully we only need 2 points on that question, right? Identify the dependent variable in the experiment. Well, the independent variable was the invertebrates, but the dependent variable is, the dependent variable is the reproductive rate of the plants, of the seagrass. Reproductive rate of seagrass. And then predict the experimental results that would support the researchers' claim that mobile aquatic invertebrates can transfer pollen. Well, you would expect there to be a greater reproductive rate in the experimental aquarium than in the negative aquarium. Four points. Easy enough. Question 4. Question 4 is about bedbugs and how bedbugs are resistant to this beta cyfluthrin pesticide. Okay, it says identify the control strain in the experiment. Well, if we look at here, this one was the one where they didn't delete anything. So the control strain was strain 1. Identify. Okay. And justify the claim that ABC8 is effective at providing resistance to beta cyfluthrin. Okay. Well, we go to ABC8 and we notice that in strain 3 it's deleted, right? And strain 3 has statistically significant differences in survivability from strain 1, right? This was like percent surviving. Not surviving. Okay, so we can say that strain 3 had ABC8 deleted. And has statistically lower survival rate than strain 1. So that would be a point. Okay, so 2 points on part A. Then part B says P450 encodes the enzyme that detoxifies insecticides. ABC8 encodes a transporter protein that pumps insecticides out of the cell. CPS encodes external structures, blah, blah, blah. So CPS is stopping it from coming in, whereas P450 and ABC8 fix it once it does come in. Explain how the deletion of P450 and ABC8 results in a lower survival in bedbugs compared with the deletion of CPS only. Okay? Well, when you delete CPS only, that's strain 4, right? So strain 4 had high survival because they could deal with the pesticide once it entered the cell. And then strain 5 had low survival Why did it have low survival? Because even though it prevented some of the pesticides from coming in, it didn't prevent all the pesticides, and it couldn't deal with it once they got in, right? Because it could not prevent 100% of pesticides, pesticide entry. Whoa. And could not deal with any pesticide. Once entered. Yep. So that was this talking about the low survival and high survival. You probably could have actually mentioned the survival rates, but it didn't ask you to mention survival rates. So like, I don't really think it was. They shouldn't give you they shouldn't require you to do that to get a point, because it didn't say specifically to cite survival rates, right? So that would be 1.4 explaining why strain 4 could still survive, and 1.4 explaining why strain 4 or strain 5 did not.
If we had the exact same mistake, but we did include the numbers with research,
You didn't, or did include the numbers? Maybe. It'll just be up to your grader or up to your reader at that point. All right, birds, cockatoos cuckoos. Cuckoos, not cockatoos. Cuckoos and warblers, right? Describe the symbiotic relationship that exists between the cuckoo and the warbler in an environment without predators. It says that without predators, the warblers were more likely to be successful if it only had warblers in the nest and not warblers and cockatoos. And that means that the relationship is parasitic. And that means that because the warblers were harmed and the cuckoos were helped. Okay. For part B, so that was 1 point for that. For part B, it says, on the template provided, draw bars in the appropriate locations to predict the relative probability of success for the nest in the presence of predators. Okay, so basically you just switch them. So if we go from warblers only and we add cuckoos, then they just go to the warblers and cuckoos. And then if we remove them, then you just go to only the warblers. Yeah. Seems really easy, but that's all there is to it. And then for C, it says, identify the symbiotic relationship that exists between the cuckoo and the warbler in the presence of predators. Well, if there are predators around, that means that they were more likely to live, because that meant that the cuckoos gave the stinky smell and the warblers provided food. And so that is mutualism. And it was just an identify question, so you didn't really need to say anything other than that. So this is question 5, which is only worth 3 points, right? 4 points. Sorry. Yeah, so this would be 1 point, 2 points, 3 points, 4 points. Okay, question 6. Question 6 says, we've got this CFTR protein that normally is a gated ion channel, and it says to draw arrows to describe the pathway for the production of a protein. Now, this question, I feel bad about it because I feel like I led you guys astray here. If a protein is going to be used in the cytoplasm within the cell, it gets made on a free ribosome and then gets released into the cell, right? This protein, even though it's being used by the cell, it is not being used in the cytoplasm. It is being bound into the plasma membrane. And so in this case, it's going to have to go through the normal protein pathway. It's going to have to go from the nucleus to the ER, to the Golgi bodies, to the plasma membrane. Did you? I feel so bad about that because we hammered home that this was for proteins that were for export, and that if they were being used within the cell, they would be in free ribosomes, but we didn't talk about ones that would have been bound into the plasma membrane. And so if they do get bound in the plasma membrane, they are included in a vesicle, and that vesicle then fuses into the plasma membrane, and that's how it gets there in the first place. That's how it stays embedded. So that would be the path there. B says identify the most likely cellular location of ribosomes that synthesize the CFTR protein. Well, those ribosomes are going to be bound ribosomes in the rough ER. Bound ribosomes. So that's 1 point there, 1 point here, and then C says, identify the most likely cellular location of the mutant proteins. Well, there's no reason to believe that it's changed at all. So where were the original proteins found? In the plasma membrane? These should be found in the plasma membrane too. Because all it did was affect some ATP binding site. It didn't affect the signaling to say where it should go. So into the plasma membrane. That was another point. So there's our 3 points for question 6. Question 7 is about the fish that changes its gender based on temperature. Okay? And so the important things to note for here, it says that there's all kinds of different phenotypes, right? So here are the males. The males are Z star, Z star, Z star W, ZZ, and Z star Z. And the females are just ZW. That's the only possible female offspring. So question A says, predict the percent of phenotypic males among F1 offspring in the cross shown above, the cross in the Punnett Square. Okay, so this is a female, and this is a male, and this is a male, and this is a male. So that is 75% male, which is what they wanted you to get, right? Predict the percent phenotypic males, 75%. All right. At least one Z or Z star chromosome are necessary for survival of the fish. A researcher crossed 2 fish and observed a 2-to-1 ratio of males to females among the offspring. So this is a little tricky, 2-to-1, because normally we're used to seeing 4 total offspring, but we're seeing 3 total offspring here. So why is that? That's because one dies. Right? So how do we get a dying offspring? Well, we go in here, we do a Punnett Square. We know one of them had to be a female, right? And the other one was Z star W. And so what that did was it gives us Z star Z, that's a male. It gives us ZW, that's a female. It gives us Z star W, that's a male, and it gives us WW, which is a dead fish, right? And so there are two males here. This is a male and this is a male. And then there's a single female, so 2-to-1 ratio. And then the question is, why is this bad? It says describe one fitness cost here. Well, 25% of the offspring die, right?
I think I wrote something like that.
25% of offspring are WW, which results in death. So 1 point here, 1 point here, and 1 point here. Actually, this was the identify, right? But I'm sure if you drew that Punnett Square, it would be fine. But you should have said that the male would have been Z star W. And then for question 8, it's talking about acetylcholine. Describe the difference in structure and function between ACR type 1 or ACHR type 1 and acetylcholine receptor type 2. Well, you can see that type 1 is going to respond to both acetylcholine and nicotine, and type 2 only to acetylcholine. And so we can assume that the binding sites are different shape. Right, so we can say type 1 and 2 have different shape bonding sites. I think it says binding. Yeah, bind, so we should say binding sites. You could have said active sites here, probably, that would have been fine.
What if you just said like type 2, like nicotine can't bind to it, and then type 1?
I don't know if that's enough. Because it says describe the difference in structure and function, right? So different shape binding sites causing type 2 to not allow binding of nicotine. So structure gives us the 1, so different shape, and function 2. So you'll probably get your function point, but not your structure point. B says, describe the effect on inhibiting acetylcholine esterase in the muscle cells that have ACHR type 2. So that's an enzyme that breaks down acetylcholine, which means that if you inhibited it, the acetylcholine would stay in the synapse, and if the acetylcholine stayed in the synapse, that would result in muscles that were continually firing or firing involuntarily. Muscles contract constantly, involuntarily. Let's point there. So I don't think we finished our tally for point 1. Let's see, or for question 1. So we had 1, 2, 3, 4, 5, 6. Predict that the white mouse, so that was 7, and then the justify 8, and then selective pressures 9, and then the 0% 10. So 10 points there. We did 10 points on 2, counted those. 4 points on 3. 4 points on 4. 4 points on 5. 3 on 6. 3 on 7. And 3 on 8. That's all the points. All the points possible there.